See, my goal of our program is to improve food security, especially in developing nations. And so we're involved in field work on 5 continents, and our output is really new varieties of crops. So it involves breeding and genetics and physiology and agroecology and even what happens in villages in Africa when they adopt new varieties, all kinds of stuff. Alex didn't want me to talk about that stuff today. He said there's people here who aren't even biologists. He wants me to focus on the modeling aspect of what we do. Modeling has been a useful component of our work. And so what I've tried to do, I'm not going to present to you kind of a coherent story about our science. It's going to be little samples of how we've used modeling to help us understand what's going on, okay? And so here's the issue. 850 million people hungry today. It's going to get worse in the future, and this is one of the big problems. Not enough water. Drought's the biggest limitation to crop production worldwide. It's going to get worse. We don't have time to focus on this. This is a very scary slide. There's no way we're going to support 10 billion people in these future scenarios that the models are predicting. Nitrogen's another big problem. Some countries like China using way too much nitrogen, causing lots of pollution. Many countries like in Africa don't have enough nitrogen. So this is another limitation to crop production. Phosphorus. Any red color here is not enough phosphorus for plants. Huge limitation to life on Earth. We're going to run out of the high-grade phosphorus ore deposits before we run out of petroleum. So here's another challenge. Though, you know, I guess the plan is these meetings, half of you are young and half of us are old. Notice how I say us and you. You youngins are going to have to deal with a lot of very serious problems by the middle of the century. And so the 2 crops I'll be talking about is this one, common bean, most important food legume on Earth, and maize, the most important crop on Earth, an important food crop in Africa and Latin America. And so here's a typical bean and actually maize production system in Latin America. It's a low phosphorus environment. This plant is very stressed. And so what happens with phosphorus is it's diffusion limited in soil. So it moves very slowly to roots. You can see here is a sim root model of a bean plant, and then these little ghost zones around the root are zones from which phosphorus has moved to the root. So it's very slowly moving in soil. There's a lot of soil here that's not supplying phosphorus to the plant just because it moves too slowly. And because phosphorus moves slowly, it ends up in the topsoil over time. And so what we discovered after some years is that bean plants that had roots in the topsoil got more phosphorus than plants that did not have so many roots in the topsoil. It's topsoil foraging. And here's some, increases in yield that we get from changing these traits. Here's one of the traits we noticed real early on is the growth angle of the main axial root of this species called basal roots. Either can be shallow or deep, and this then is better for topsoil foraging. And you can see here genetic variation in this trait leads to a 6-fold increase in phosphorus uptake in the field. However, as you can see from this study in Honduras, there's trade-offs. In this case, genotypes that have shallow roots do better under low phosphorus, but genotypes that have deep roots do better under drought. Obviously, water tends to be deep in the soil under drought conditions. So there's a trade-off here. It's one of the concepts I want to introduce is that in biology, life is full of trade-offs. And so, in fact, here's one of our types of models. This is a simple optimization model some years ago where we showed that the optimum growth angle of these axial roots was a function of how much phosphorus stress you had and how much water stress you had, very obviously. A more interesting model that we developed was actually taken from game theory because, as somebody mentioned, I think it was you, Dan, something about plasticity, that roots can change in response to their environment. Not all of them, but some of them, some genotypes can. And so we thought of a situation where, okay, well, in a tropical farmer's field, there's low phosphorus, yes, but there may be drought, there may not be drought. That's stochastic. And the plant can just be stubborn and do the same thing, or it can respond to the environment. And what's the optimal strategy? And you have to think about what your neighbors are doing. Because if you're doing something that's different than all your neighbors, you have a competitive advantage. And so we constructed a very simple game theory model where we had non-plastic plants that were always the same. They were deep. Plastic plants that could get shallow in response to low phosphorus, which does happen in some plants. And then we had a stochastic distribution of rainfall, and we had neighbor effects. So here's what happens with drought frequency. Over each one of these time periods is a whole season, and then the fitness of the plant resulted in more offspring that would lead to the next season. And so we ended up, if we had frequent drought, it was better if the plants just were not plastic and just stayed deep, always were deep, because that was the best strategy. But if we had less drought frequency, the plastic plants tended to dominate because they could take advantage of this topsoil phosphorus. That's very simple. And when you think about spatial competition, the fact that these guys are competing, and we've shown in modeling and in field work that they actually do compete differently based on their growth angles, you could say, well, in that case, there was a stable equilibrium that was set up between plastic and non-plastic plants, because if all the plants in the population did the same thing, they were all competing with each other. So there was always a niche there for plants that were doing something different, and that gave us kind of a stable equilibrium between plastic and non-plastic plants. And then combining the both, the drought frequency here, low or high, and the spatial competition, it told us that, well, under conditions of high drought frequency, most of your plants are stubborn, they just go deep, but some are plastic. In a wet year, these guys really, this is a good strategy, and they have a different niche than these. But if we have less drought frequency, you're better off being plastic. Occasionally just go deep, and you have this release of competition. So here's a very simple game theory model that helped us understand how we could improve these cropping systems. In this case, can we breed plants? So here's kind of the take home. If we have increasing drought frequency, and here we have fraction of plastic plants, you want less plasticity as you have more drought. And this, because there's genetic control for angle and also plasticity, we're able to construct, in this case, multi-lines, combinations of plants that have different strategies in the same field. And this is not a model, this is actually field work in Honduras. Amelia Henry went out and planted these side-by-side plots of your shallow, deep lines. And this is the yield, and you can see some very stressful environments in farmers' conditions in Honduras, up to some pretty decent environments. And then the red is the multi-line, where the seeds were mixed together. So the farmer was treating them as if they were one genotype, even though it was two different phenotypes. Phenotype is what the plant looks like. And you can see there's a consistent yield advantage to having these mixtures. In this case, this concept was informed by our modeling of how plants interact and how they optimize for two resources. As another, I won't go into detail here, we don't have too much time. Another strategy is, in this case, we have one whorl, one set of basal roots, in this case 2, 3, 4. And this is SimRoot, takes into account the costs and benefits of everything. So as you make more roots, they cost more, and then they branch less and they grow less deep and so on, and we'll come back to that later. But this model did show us that as this is under genetic control, as you go from 1 to 2 to 3 to 4, there's an optimum here where now you can capture both shallow and deep resources and it kind of co-optimize those two contrasting spatial resources. And this is under genetic control and has been very useful in being breeding. Here's another trait. Now we're looking under a microscope at root hairs. These are subcellular protrusions of epidermal cells that go out into the soil. Because phosphorus moves slowly in the soil, this is very helpful in extending the zones of depletion where the plant can access soil phosphorus. This is the result of scientific breeding. This is a landrace grown for centuries by people in the highland of Peru. And so here you can see variation for that root hair length is strongly related to phosphorus capture in the field. Here's the field. You can see a very nice relationship. Longer root hairs, 250% more phosphorus capture. So it's a nice target for breeding. We wanted to understand and unpack this a little bit, though, and here we used modeling. This is SimRoot modeling, which is a structural functional model. And so this is actually a Arabidopsis is what we did this with, which is a little kind of mustard plant that plant biologists like to play with as a test organism. So this is what a Arabidopsis does. If you give it high phosphorus, it doesn't make very long or very many root hairs. If you give it low phosphorus, it's plastic. It says, hey, I need phosphorus, I'm going to get long, dense hairs. This is what nature does. And then using the model, we created two things that nature never does. In other words, we can make them denser but not longer, or we can make them longer but not denser. And the model also allows us to estimate pretty precisely how much phosphorus we got because it's a simple diffusion process, easy to model. So you can see as we increase the diffusion coefficient for phosphorus, we get more and more phosphorus capture. But you can see how closely related it is to the actual root phenotype. And so what this model showed us is, yeah, you can improve length and density and then other traits that happen with root hair phenotypes. And their combined effect is much greater than you would think. Here's the additive benefit for having these traits for phosphorus capture in blue, and the red is the actual synergistic benefit, the combined benefit. In other words, these traits are synergistic. And here's another concept I want to introduce to you non-biologists. There's a lot of synergism in biology. It's good to have an upper jaw, but especially if you have a lower jaw, you know. So in this case, it's good to have long hairs, but especially if you have dense hairs, look at the benefit. So in this case, our benefit from having all four of these traits was over 400% what you'd think from the additive benefit. Now this was important in informing breeders because breeders often think in terms of additive, like, oh, I've got X is good and Y is good, put them together, I've got X plus Y. No, you might have X times Y. So this was useful for guiding breeding. And in fact, here's a synergism study from the field in Mozambique. In this case, it's root hairs and shallowness, the two traits I showed you. So the baseline is short hairs and deep roots, not good for phosphorus. This is a low phosphorus field. If we have shallow roots, we improve growth by 60% in the field in Africa. If we have long hairs, we improve growth by 90%. You'd think if we had both together, and these have the same genetic background, we'd have a benefit of 150%, but what we actually saw was a 300% benefit because those hairs are better on shallow roots in the topsoil than they are on deep roots. Now, one reason I'm picking these examples is because I think there's a lot of interest in among biomathematicians in shape and form and stuff. I'm trying to get to function here, and there's some kind of perhaps unexpected or emergent properties of form when you think about function. And here's an example very clearly showing you this synergism from two different traits. And so this is not modeling, just to show you these things have been useful. Celestina Joshua is a bean breeder we work with in Mozambique. She's just released 15 new lines that have between 50 and 150% better yield using these three traits I just showed you. So the modeling was quite useful in guiding the development of new crops that are having impact in Africa. Now I'm going to tell you a different story about maize, corn, and nitrogen. This is SimRoot showing you the pulse of nitrate leaching down through the soil in the spring with rainfall. And you can see the zones where the nitrate has been taken up. Nitrate, unlike phosphorus, is very mobile. It moves. And that's the problem. Estimates are that crops only capture less than, or about a third of what we apply. All that, the 2/3 of nitrogen we apply to crops in the US and Europe, it isn't taken up, it just causes pollution, pollutes the air and the water. And once again, in Africa, they don't have nitrogen fertilizer. So we're trying to develop a root system that can catch this stuff better as it leaches down. And so we developed this idiotype, and idiotype is like a conceptual model. It's like our goal of what we would like. If you're breeding German Shepherds, you have an idiotype of what a German Shepherd should look like. And same for corn breeding. We have a goal in our mind of what the corn plant should look like. So here's our idiotype for corn capture of nitrogen and water called steep, cheap, and deep. And there's a bunch of traits. The idea is to try to get as deep as possible, as fast as possible to catch that stuff. I'll give you a few examples. Optimum number of roots. So here's natural genetic variation. Few of these nodal, these are the axial roots in this species are called crown roots. And here there's many. This is genetic variation. And now SimRoot, these are Seminole roots, which are different. Here's the primary root. These are the nodal roots we're talking about right here. These are the crown roots. So this one's only going to make 6, and this is going to make like 30, which is what we see in nature. And SimRoot was very useful here because this is an idea that actually came directly out of the model that we didn't expect to see this. We were actually simulating something else, and the model was telling us, hey, you know what? This is important because you make so many roots, they're competing with each other because nitrate moves, and so they're wasting effort. They're all competing for the same nitrate. They're using up plant resources, and therefore they cannot grow very deep. And this is actually a better phenotype, according to the model, which is a little counterintuitive. Less roots, better for nitrogen capture. This is what the model says. Now, what we're seeing here is kind of this reflected roots because in a cornfield, there's maize plants very close together. So rather than try to simulate 6 plants together, we just have one plant and reflect the roots, representing the roots coming from the neighboring plant. So you can see this is a phenotype with 46 nodal roots, and here's one with only 6, and you can see this got deeper, and it has more branches. SimRoot is a structural functional model, so it knows if I have less roots, I've got more resources to devote to the roots I've got, and so they can go deeper. And so the model is telling us that if we decrease nodal root number, we'll have we have a big effect on shoot mass under low nitrogen. We were actually simulating this. This was the object of the study, which didn't have a very big effect. So this is a case where the model really gave us an idea we didn't have already, which is, wow, the big difference here is in how many roots we make. And so this has actually worked out. Of course, we don't believe models, especially breeders, very stubborn, skeptical people. So we have to show it in the field to convince a breeder to deploy these traits. So here's a study in South Africa with low and high nitrogen, and basically confirmed exactly what the model predicted. By reducing the number of crown roots, we improve yield significantly. We get an 180% increase in yield under low nitrogen. This is under drought. We get a 60 to 116% increase in yield under drought if we decrease the number of nodal roots. Here's another example I want to show you. Few long laterals, the same kind of concept. This is the phenotype we're looking at now. This is one nodal root now, and these are lateral roots. And you can see this is natural genetic variation. This one made a few roots, and each root then has a lot of resources, and it can get long and thick. And this one makes more roots that are then competing with each other. Now, this is what this simulation looked like. This was a simulation done with part of the PhD thesis of Johannes Postma, who's at Forschungszentrum Jülich, where Hendrik comes from. And so here's the few long phenotype, and here's the many short phenotype. And so the model knows. The model is smart enough to know that if we make fewer roots, they have more resources and they grow longer. So we didn't specify root length here. The model predicted that these roots would be longer, which is actually what happens. And the model also predicted that if you increase nitrogen, that the number of lateral branches, the optimum number of branches is, let's say, around 8. And for phosphorus, we have a greater number of branches. So it's basically saying that this phenotype is better for phosphorus because you have to get closer to the resource. This one's better for nitrogen because it's moving around, so we just want to explore the soil very extensively. So this is an insight provided by modeling, and now once again we try to confirm this in the field. Here's under low nitrogen. It turns out the few long phenotype has 32% more yield under low nitrogen, and under drought you can see here as we decrease the lateral branching density, here's our yield. We can get 61% more yield under drought by having a few long phenotype. So here's an example of a different kind of trait, high RCA. What is RCA? RCA is root cortical parenchyma. Here we're looking under a microscope. This is a laser ablation image of a maize root. It's not a model, it's an actual root. And this green area here is airspace. So living cells have been killed and converted to airspace in the formation of this tissue called parenchyma. Okay, and it makes the roots a lot cheaper. Here's natural genetic variation. Here's a plant that doesn't make parenchyma. All the cells are still there. Here's one that killed a lot of those cells and turned them into air spaces. Now this is what we thought. This might be a good thing under nutrient stress or water stress because now your roots are cheaper and can go and explore more soil. And so this was Johannes' research here for his PhD thesis. Here's a SimRoot model showing a heat map of where parenchyma is formed in time and space, and we had to obtain this map by measuring a lot of plants. And you can see it's dynamic in time and space, and then we also know how it affects respiration and nutrient content and so on and so forth. So the model's able to. We also know nitrate uptake kinetics in the root system. We have a map of the maximum rate of nitrate capture and the affinity of the root for nitrate. These are non-uniform. That's one of the points I wanted to show this to remind me to emphasize that roots and leaves, any kind of biological system, is highly non-uniform. So we can't just think of this as an object. That's got a lot of interesting dynamic properties here. And so using all this information, Johannes, here he has a simulation, a maize plant without parenchyma and one with parenchyma. They're growing in a low phosphorus soil. The model includes the shoots and canopy photosynthesis. It's just not being visualized. And you can see over time the one with the parenchyma grows better because every unit of root is cheaper, can grow a little bit more, capture a little bit more phosphorus, there's more photosynthesis, there's more carbon. It's an autocatalytic feed-forward benefit, so a relatively small difference in cost results in a large difference in growth. And so his model predicted that if we have unstressed plants out here, and if this is increasingly stressed by, in this case, lack of nitrogen, the benefit from parenchyma increases. In fact, the model predicted you could get a 60% more growth by having parenchyma under low nitrogen, also phosphorus and potassium. And I actually didn't believe it. I think I ended up giving Johannes a bunch of nice Dutch cheese and to apologize for having doubted his results, because it seemed to me it was like, wow, this is way too good. How could parenchyma improve growth that much? But when we went to the field, we actually saw that it had a significant effect. So here again is a low nitrogen field we have in South Africa. Plants with and without parenchyma. The plants with the parenchyma had 58% more yield, which is almost exactly what the model predicted. Under drought, it was even better, 800% more yield, because in maize, if you do the drought stress right, then they don't even pollinate correctly, and you can have a tremendous effect on yield. So here, plants with more parenchyma under drought look a lot better than those without parenchyma under drought. And that's recently been confirmed in Africa. We did some field work in Africa showing this is really a useful trait for drought tolerance. And these traits are being deployed in African breeding right now. So one of the things I wanted to bring up, coming back to this synergism point, here's a heat map of parenchyma formation in a maize root at 42 days after planting. And now we're going to zoom in on a different scale. This is a different model called root slice, structural functional model of cellular, subcellular processes. And you can see an parenchyma lacuna there that we're zipping through. And these things are probably going to interact. And in fact, they do. So this is a modeling study looking at the interaction of those two things I showed you. One is how many roots we form, and one is do they have a parenchyma. And you would think that it's more important to have cheap roots if you have more roots. And that's what the model tells us. So here's a number of roots, and here's the parenchyma formation. Here's the biomass under low nitrogen, and there is an interaction. This is just not a plane. It's a complex shape. It's a complex function. And if we increase nitrogen, that function changes, obviously. As nitrogen ceases to become a problem, it doesn't matter how efficient you are at getting nitrogen. Same for phosphorus. They interact, and if you change phosphorus levels, the interaction changes. And I think this is obvious to biologists. It may be obvious even to mathematicians. So if we have, the problem is we have n phenes, a phene is an element of a phenotype. Existing in X states, they interact to create X to the n permutations. So example, in our lab, we're looking at over 24 phenes, and you might say there's 3, 4, 5 states of interest between shallow and deep, or even 2 states on or off, and you end up with a very large number. And there's no way we can do field work with millions of plants, even if we could, generate them all. We couldn't afford to do field. So modeling becomes very important. And the approach we've taken then is optimization modeling. So this is a genetic algorithm, a genetic optimization, where we have a bunch of a decision space with our phenes here that we can vary, and then we run the model and see how this combination of decision variables affected our objective variables, which is how much nitrogen and phosphorus capture we got and so on. And it tries to find the Pareto optimum of situations where you're maximizing this and minimizing something else and finding the best combination. And so we're using this is a genetic algorithm. It kind of simulates what happens in biology, where you have the number might be composed of 4 different elements, like different genes on a chromosome, and then it recombines them. The algorithm recombines them in random ways and tests the fitness at every step, and only the best offspring are allowed to proceed. And so it kind of simulates evolution. And this is the way to explore, because in this case, the phene space we're interested in, here's low nitrogen to high, low phosphorus to high, and here's growth of corn simulated. There's actually 10 to the 23rd scenarios of interest. That's a large number. But obviously we don't have to look at every one. Using the genetic algorithm and the optimization algorithm, we just look at a small subset and ignore the ones that are clearly bad. But even at this, it took 1 million hours of CPU time. And so this is really interesting, and we're finding cool stuff, and we don't really know how to analyze this, because these tools were developed in engineering where you're trying to find the best solution. In biology, there is no best solution. There's a bunch of co-optimizing, solutions that all seem to work. You look out in nature, you don't see that plants all look the same. They look different, but they all work. So we're not sure what to do with this, but it seems like a good way to proceed in analyzing this phenome space. Another challenge we're confronted with is other organisms. In the U.S., a field of corn is all corn, but if you go to Africa, they grow corn and beans and everything together. And so I wanted to mention this study we did with the 3 sisters, maize, bean, and squash, which is a very ancient polyculture grown by the Native Americans who used to live here before Europeans came here. And society was based on these 3 crops. That's why they called them the 3 sisters. And so here they are growing together. maize, squash, and bean, and these wonderful, I mean, this is a complete diet nutritionally, low glycemic index. It keeps for months without needing refrigeration. And also agronomically, this beautiful system because the corn is a stalk, the beans climate and fix nitrogen, the squash covers the ground. It's just an amazing system. And in fact, here's a, it was recognized on a U.S. currency. Our American Thanksgiving holiday actually comes from the Native Americans who would give thanks to the three sisters. It's actually not a religious holiday. It's thanking three plants. That's what Thanksgiving is all about. And so this, again, is work we did, part of Johannes' PhD thesis. So you can simulate over time these three architectures of these three species. You see here the three different colors, and we know where the nitrogen is and the phosphorus, and it's dynamic. And we can look at their interaction in a way that would just be impossible to do with real plants. And this study was interesting because it showed us that having the three together in the same spot as opposed to a meter apart made it much more efficient. There was niche complementarity. These three occupied the spatial-temporal exploration of the soil when these three are together is more efficient for nitrogen than if they're separate. So it's even a more beautiful system than we even knew about. But for phosphorus and potassium, because they move so slowly, these don't interact. So that was interesting. And once again, that was useful in guiding our field research because maize and bean are grown together, and it told us that for phosphorus, which is a concern in Africa, we didn't really have to worry about the fact that this maize root is totally dwarfing these two bean roots because the model said the interaction is just negligible because the roots, even if they're 2 millimeters apart, they're really not interacting for phosphorus. So this gave us some useful insights that was helpful in guiding our breeding work for Africa. So let me summarize real quickly what I wanted to say. Function is important for one of the principal challenges facing our species, how to feed ourselves on this planet. And root structure is important, but it's not simply related. The relationship of root structure and function is not simple. There's internal complexity. I talked about root cortical parenchyma, for example, or nitrate uptake kinetics, showing you how non-uniform a root system is. And then there's the environmental complexity. I talked about, for abiotic resources, how immobile phosphorus is versus water and nitrogen, which are very mobile, and that totally changes what's useful for a plant. And then there are things I'm not talking about today. Well, I talked a little bit about other plants, competition with other plants. And then things are eating roots, and the microbes live on roots, so it's a hugely complex challenge that beyond our capacity to explore through empirical research. I think we really need modeling to come to terms with this. And I gave you some examples of what we're doing as kind of a microeconomic analysis, looking at a plant through a microeconomic lens and saying how can it optimize performance. And we've used cost-benefit analysis, I showed you, trade-offs, I talked about optimization, I showed you, and some game theory have all been useful approaches towards this. So, functional structural plant modeling has been critically useful in our work. And, models integrating plants, soils, microbes across scales, from genes to ecosystems, will be an increasingly important discovery platform in years ahead. I'm very pleased, Alex and Dan, that you've gotten us together because I think this is an area that's going to be very, very important and a scenario in which the United States is very much behind Europe and China and Australia at the moment. And I think you youngsters in the room are going to see more and more people, even genomics, whatever it is, they're going to be using powerful in silico tools to help contextualize their results and scale them to say how this process at this scale is going to affect other processes at other scales. Thank you very much.